Yep. Send them in. Hi, Mrs. and Mrs. Abernathy. I'm Dr. Edwards. I just want to reiterate there's nothing to worry about here, but shall I explain what we're going to be doing today?
It's kind of hard not to be nervous when we're about to find out if our kid will be born with a genetic disorder or not.
To be clear, you'll find out your results in about a week. Testing takes around 2 to 3 days. Results coming in soon after that. Essentially, what we're doing is checking to see if the embryo, which we have just obtained from you after completing your IVF sessions with your donor, of course, to fertilize your egg, has any chromosomal or genetic disorders.
Could you maybe explain a little more? I'm a little out of the loop.
Of course. Let me see. We will grow the embryo in a special lab for around 2 to 3 days. Then once it is split up into 8 cells, your embryologist, me, will take, let's say, 2 of those cells to do special testing to see if it has a disorder of any kind.
Don't worry, all we're doing today is finalizing the IVF process.
And we'll find out in a week?
Ish. IVF, pre-implantation genetic diagnosis, yada yada yada. Choosing your genes? Hi, Mrs. and Mrs. Stern. I'm Dr. Edwards. I'll be your doctor today. I just want to reiterate there's nothing to worry about here. All right?
Please, go ahead. We're excited to be your,
Guinea pigs, dear. We're her guinea pig.
Call it what you will. We're about to make science history here.
Well, alright. Can you just explain, please?
Surely. Well, a while back, this procedure called IVF was discovered, and it's basically a way to fertilize a woman's egg with the man's sperm, your donor, in a test tube or in vitro. Then the cells combine to one cell called a zygote, and it divides and turns into an embryo. Then the embryo gets implanted back into the woman's uterus, and yeah, so IVF kind of inspired this other procedure. It's called pre-implantation genetic diagnosis, PGD for short.
and what's that?
I'm getting to it, Mrs. Stern. PDG starts off with the normal IVF procedure. However, once the embryo has developed for around 2 to 3 days and is then comprised of around 8 cells, the embryologist at this point, me, would take out 3, let's say, of those cells and scan them under a microscope and some other things to test for any chromosomal or genetic disorders. And now comes to the fun part. Years ago, when I was doing this relatively simple procedure on a couple, just like yours, I figured out a way to change science forever. Instead of testing for disorders, I would test for the genetics of that future child, and instead of just observing them...
You can change them.
Exactly, Mrs. Stern. I could change the genetics of that cell to the parents' pleasing.
And is that dangerous?
As is everything, but we won't know until we have sufficient data.
And that starts with us.
Your guinea pigs.
My guinea pigs. Hello?
Hi, Dr. Edwards. It's Mrs. Stern. My wife and I met with you this morning.
Ah, yes, Mrs. Stern, my guinea pig. How may I assist you?
I just wanted to clarify something on how you're going to be doing all of this, you know, the PGD stuff.
Ah, yes, that I can do. Okay, so remember how I was saying I'm going to take 1 or 2 cells out of the embryo?
Yes.
So I suck the cells out through an extremely tiny needle.
Okay, so that sounds a little dangerous. Then the embryo would only have around 6 cells instead of 8.
Very, very true. However, the embryo is capable of recovering lost cells by growing back new ones.
Ah, okay.
Right. So the next step is DNA extraction, and there are a few different ways to complete this task, but my favorite, which is what I'll be doing today, is boiling the cell.
Boiling?
Yes, boiling until it pops so I can extract the DNA from it.
Oh.
And let's say we want to check using PGD for if your child would be prone to cystic fibrosis. What we would do is look for the CTFR gene, which has the information to produce a specific protein. And if the DNA sequence is not in order, then the gene cannot properly produce proteins causing cystic fibrosis. So to check if the DNA sequence is correct, we use something called PCR, which duplicates the gene, and the question is how many times you would like to right. So you can take a closer look at the gene.
Oh, okay. I get it now. This is all PGD, correct?
Yes, this is all PGD, and that's what you wanted an explanation on, correct?
Yes, you're right. Thank you, Dr. Edward.
You're welcome, Mrs. Stern. See you tomorrow so we can discuss what you want your child's genes to be. All right, see you then.
I want our child to be handsome.
And perfect too.
Brown hair,
Red hair,
green eyes like mine,
intelligent.
Harvard material, and a basketball star too.
I think yes.